Hi, my name is Megan Rokup, and I work here on MIT campus at the Broad Institute of MIT and Harvard, which is a biomedical research institute. Today, we're going to do a lesson talking about using DNA to identify people. And as you can see on this slide, I'm showing you a picture of DNA.
So this is what DNA looks like.
This is what DNA looks like. It's a molecule made up of atoms. There's a lot of carbons and hydrogens and nitrogens and oxygens. And it forms a three-dimensional structure shown here as a double helix. Now, the backbone of this structure, which is the two spirals you can see, are always the same along the DNA molecule. But the rungs in the middle can be different. And there's actually 4 different kinds of rungs, and we abbreviate them A, T, G, and C.
Is this DNA how it actually looks like?
So while this is what DNA actually looks like, a three-dimensional molecule, when the PCR reactions are done and I have my segment copied, is I'm going to transfer, again, a small amount of that into these tubes right here. And these tubes, I'm going to mix the DNA with an orange dye. This dye is going to allow me to see the DNA, and then I'll be loading it onto this gel here. So the first thing I'll do is transfer a small amount of orange dye into each of the tubes that I'll be placing the DNA samples into. You can see this orange dye in my pipette tip. It's just a colored substance that we can see with our eyes. DNA is transparent, so we can't see it with our eyes. This is what DNA looks like. So while this is what DNA actually looks like, DNA is transparent, so we can't see it with our eyes. And you can see that the orange dye is almost at the end of the gel here. So this gel is almost done running. And when it's done running, it'll beep to tell me it's done, and then we're going to take it into the darkroom so that you can see the results. So here we are at the darkroom. This is where we're going to visualize the gel. I've got my gel here, and we're going to go in and shine a certain wavelength of light on it so we can see the DNA. So here we are in the darkroom, and I've got my gel. I put my gel into this light box right here, and then I simply turn on the light, and all of the bands of DNA appear. This is what DNA looks like.
So this is DNA that you can't see because it's transparent.
DNA is transparent, so we can't see it with our eyes. So here you can see the results of the gel.
That DNA helix looks pretty similar to that orange dye. All CGI, artist impressions. DNA. Same with RNA, of course. It's all drawings. How do we know it even exists? Because it's transparent. You cannot look with it with an electron microscope. And if it was possible to see it with a microscope, an electron microscope, then you would totally destroy it. Because electron microscopes use high vacuum and 300° Fahrenheit electron beams that passes through a thin slice of sample, and sample preparation can take up 2 hours or 2 days. RNA, just as imaginary as DNA. Bioscience. This is just a joke. This is the lunar lander. As you can see, this is all a joke. You cannot take this stuff serious. Anatomy of a virus. They become totally crazy. Who made up this bullshit? Look all this crap. I don't understand why people buy this. Well, you must be stupid to believe in any of this science.
And remember, these two strands are wrapped around and around and around and around and around each other, right? One is going like this, one's going like that, and they're just like wrapped around. As I tug them apart to make a new strand, to synthesize a new strand, those two new double helices are so totally intertwined with each other.
DNA is transparent, so we can't see it with our eyes.
Every turn that there was in the double helix is now a twist and turn connecting the two, sort of entangling the two helices.
This is what DNA looks like. DNA is transparent, so we can't see it with our eyes.
So I have the two new double helices entangled with each other.
Yeah, we all love a good story. And you just brainwashed 25 kids with theoretical nonsense.
At the end of the runtime, the gel is removed from the gel tank and transferred to a gel dock system or trans illuminator to visualize the DNA.
Finally, we're going to see some DNA helixes.
This gel picture contains samples that are both of high molecular weight DNA as well as degraded DNA.
I love science. Piece of shit. So you extract this RNA from an invisible virus and then put it in some liquids and then stash it in your tiny heating machine, your PCR machine, which is nothing more than a heater.
Instead, we need to copy it so that we have more DNA such that we are able to see it on the gel. Is that this machine, although it's called a PCR machine, all it really is a heating block. This machine is a heat block that changes temperature really rapidly. So it's going to change temperatures, and each step of the copying reaction happens at a different ideal temperature.
And then this multiplies. And then you can also change it in cDNA, which is also theoretical nonsense. And then you can determine somebody have a virus. Let's see how the average corona test is being conducted.
Hello!
So let's put some swabs in your nose and in your throat. Hold on. The way we do it, we can have false positive tests, because science tells us aerosols can linger in the air for a long time and can land on the swab now. And then they put this swab with this lingering coronavirus in your throat. This looks like fake. Look where she come from with that swab. And now you're going to give that lady a hanky for a possible tearing eye or a dry of your nose. So you're going to accept that hanky. That's a little bit dangerous. You can get infected. Don't accept anything. Look how long she stands there with that swab in the wind. And why are you going to touch that person's head with your dirty gloves? Maybe this woman have a tiny corona particle stuck in her nose hair. And now you take the swab and push it up to the nose. That's a little bit dangerous. No need to touch the face, or is this staged? Ah, that was a quickie. Oh look, what you going to do with that hanky? It can be full with the corona that landed on it. Yeah, wipe that corona all over your face. And don't forget to put some sanitizer on your hands after the test. You're not changing your gown. Are you only protecting yourself? And your gloves are touching your gown, idiot. And you conducted all these tests today, like 30, and then you leave those in here for a full day or so. UV light makes the virus inactive. Can you tell us we have active virus or inactive viruses in us? So this woman hands you over the swab. What kind of mask is this? And how many times this person got tested? You're telling us the results coming back after 2 days from the lab? Maybe this person is infected and it's breathing on the swab, which can give a false test result, of course, for the person in the car. Watch out for lingering corona particles. Yeah, and give her the hanky so she can wipe her face. And now this nurse is breathing all over the swab and change hands. Don't tell me this one is holding her breath for the whole time. If you work in this branch and you don't understand cross-contamination, then you're not fitted for the job. Yes, here is the magician's hand again. Let's do the swab in your nose. Or not. Ah, it doesn't matter. I can't imagine why you let your touch your face by this nurse. Those people are super spreaders. Don't touch your mask if you believe in the corona. And you too, you believe in it. Stay off your mask. You're touching other stuff. What the fuck is this? That looks like a coffee filter with straps. Here, coffee filters. Actually, it's not a bad idea, because those cost 2 or 3 euro, and you have 100 pieces. You put some rubber band on it, and you have a lot of face masks. Cheap. And you can put them on your head, so you look like a corona smurf. Hey Davey, what's up, dog? Those people love those face masks. They don't even care. They don't know what they are doing. Go wash your hands, super spreader. Those people don't realize they're playing with fire, with the most dangerous virus ever. If you come across people like this, for example in your local supermarket, and they are touching their mask and also touching all the goods in the shop, then please report them to your local authorities, because those are the super spreaders. They need some re-education. about this dangerous coronavirus. You must understand, those are the ones that keep us in lockdowns. Because of this, we have a high infection rate in Holland. The super spreaders, they should be locked up and re-educated. Those are dangerous people. Please report them. Even our minister president is a super spreader. Look when he takes off his mask. So now you took off your face mask, and then your face mask need to be disposed as chemical waste because it's pretty dangerous.
Zo.
This super spreader wants us to follow the rules of the Dutch CDC, but he don't like to follow the rules himself or the advice.
Dames en heren,
This is an instructional video from the Dutch CDC, and it shows you how to take off your mask.
Je doet het masker af door alleen de elastieken vast te pakken.
We only touch the rubber straps and then pull it off. So far, so good. And now you do it wrong, because you touch the mask with your hands
Gooi het masker in een afvalzak en raak deze niet onnodig aan.
and dispose the mask in a bin bag and don't touch it anymore.
zo.
Or just put it in your pocket for later use.
Pas handhygiëne toe door je handen te wassen of desinfectie te gebruiken.
And after you dispose your mask in a proper way, you wash your hands. You're telling us this all the time. Wash your hands, wash your hands, wash your hands. Until they bleed. And I don't think this is a proper way to dispose your hazardous mask. Because the one that have to clean the bin bags is in a big danger. Especially if you don't know there is a face mask.
Wij spreken elkaar, naar planning in ieder geval dinsdag weer.
Oh shit, I almost forgot my face mask. Where I have it? Oh yeah, here in my pocket.
And
Hey Andre, you're dropping something.
and